two people who hopefully don't need much introduction, Dan Mark and Carrie Lee. They were both here at the DCRI long before the DCRI was the DCRI. Kerry's a biostatistician, one of the faculty in the Department of Biostatistics and Bioinformatics, and really one of the early founding members of the DCRI. And Dan Mark is a cardiologist, outcomes researcher with an expertise in health economics, also who was here really before the DCRI was the DCRI. And today they're going to tell us the true story behind the Tac Trial. So just briefly, Tact was a NIH-funded randomized trial of chelation therapy and some vitamins. And this is one of those alternative medicines which most traditional doctors are sure don't do anything, and yet are widely used by patients and offered by practitioners. And the NIH, together with Kerry and Dan and other investigators, took on funding a rigorous clinical trial to investigate chelation therapy. And the main results have been presented and published, but I think we'll hopefully hear some of the behind-the-scenes stories that went on during the trial during this presentation. So Dan, I think you're starting off.
Yeah, thanks, John. My mic picking up okay? I had to move it, so if I seem to fade off, it may be as I turn to the right. So I don't know that we actually agreed to tell the true story, John. We just told the story. There's many stories to be told. We're going to tell one of them.
Don't tell the true story.
Yeah, let's not ruin it. So I have the disclosures shown on this slide. I don't think Kerry has any disclosures relevant to this presentation.
And I always tell the truth.
You do. And that's why you're...
Chelation therapy shop I saw down the street.
Hey, it's a chain of shops. It's not a single shop. You gotta franchise in order to make money these days. So the way Kerry and I decided to organize this was he was going to do the blow-by-blow presentation of the actual trial and the trial results, and I was going to do the color commentary around some of the events that took place and why we did the study. So I'm going to start off by talking about things that led up to the Tac trial. Kerry's going to take over and go through the trial itself, and then I'll come back and discuss a few of the many things that have taken place since the trial results have been presented. Not all of you perhaps even know what chelation is or why there's a big fuss about chelation. It's basically, chelation is a chemical process that binds a certain type of element, metal ions, bivalent, trivalent metal ions such as lead, calcium, mercury, a variety of things that we may or may not want in our circulation. And so it binds these tightly in a six-prong molecular shape, which is shown on this slide, and allows this complex to be excreted harmlessly without metabolism in the urine. So the EDTA, the compound that we're actually studying and talking about today, was synthesized initially by a German chemist in 1938, and they didn't know what to do with it originally. There was some bizarre things that they did with it before they decided that it could be useful for clinical medicine, and the early papers show its use in lead toxicity. It had a noble start. It papers in Science and things like that. So it didn't start off being kind of a kooky therapy. So how did it go from lead toxicity to atherosclerosis? That's the question. And I don't know that can be completely answered, but there was a clinician in Detroit who, for whatever reason, was giving this compound to a variety of patients that had metastatic calcification. That is, calcification, heavy deposits of calcium in areas of the body that it's not supposed to occur in, and allegedly found that these deposits in kidneys and tumors and other things were dissolved over a period of time, that they resolved, something relatively unexpected. And he elected to try this out in 20 patients with atherosclerotic coronary artery disease. Now, they didn't know, I mean, it was before cath and all the modern tests. So basically, it's patients with severe clinical angina or myocardial infarction, all of whom are pretty symptomatic. And they took 20 patients, gave a mean of 35 infusions, each one of which was 5 grams of EDTA, which is a bit more than what was used in the chelation trial that you'll hear about, and was administered relatively slowly over between 2.5 and 4 hours. And what they observed was that the 19 patients who survived this study, there was what they called unusual symptomatic relief. And the other interesting thing was that the patients didn't have any effect of therapy until at least 20 weeks had passed. So they concluded this was unlikely to be a placebo effect, because if the patients had gotten a placebo effect, they should have gotten something relatively quickly. The other interesting thing was that the benefits seemed to persist for up to 2 years after discontinuation of the infusions. And the authors speculated, and this has been the source of much misery since, perhaps, that EDTA was actually removing the metastatic calcium from the atherosclerotic lesions, which were somehow coupled to the abnormal lipid deposits, and through that process somehow was ameliorating the coronary artery disease symptoms. A second paper that's noteworthy came from Philadelphia and was published in the AJC in 1963. So these investigators took a small cohort of 10 patients following up on the study of Clark, and they gave them 20 infusions of 3 to 4 grams of EDTA. They saw nothing in any patient. They discontinued the program. Basically, they were ready to pack it up. And then 6 to 8 weeks later, the patients started coming into their visits describing dramatic improvements in their symptoms and spectacular reductions in the use of nitroglycerin. And so that got everybody's attention. And so they decided to do an additional 28 patients plus 9 patients in a double-blind crossover study. Now, the difficulty that they have here is they really have almost no tests to figure out how to follow the patient. There's no biomarkers, there's no cath, there's no echo. I mean, how do you actually figure out whether the patient is better or worse, the same? So they were mostly just following the patient's symptomatic status, and what they decided was that in the second cohort, and combining the two, there was about a 2/3 improvement in symptoms over time, but that the effects did not appear to last, and that the prognosis did not appear to be improved. Many of these patients actually died during the course of this study. So they concluded that chelation, while having some interesting properties, was not actually a useful clinical tool for coronary artery disease. So somehow between that and the 1970s, chelation became what's called alternative medicine. The mainstream interest faded, but a group founded this American College for the Advancement of Medicine, ACAM, to promote the use of chelation therapy. And they created this standardized regimen that you'll hear about more, that was actually the one tested in the Tac Trial. Now, the problem is they pushed, instead of pushing more on the clinical series, as you'd seen before, they pushed from an intuitive, non-experimental perspective that this made a lot of sense and therefore it must be right. And that, combined with a lot of anecdotes, was basically how the field was driven up until the start of TACT for the most part. So lots of testimonials, books, very few papers. Most of the literature is case reports and case series. Incidentally, this book was endorsed by Linus Pauling, who was reeling from his failure with vitamin C and decided he would go for a definite winner. So So this is the kind of thing that you got in this literature. This is a chelation website. And the guy says, I've seen marked improvements in high blood pressure, angina, leg cramps, diabetic leg ulcers, improvement in mental alertness and memory due to improved blood flow in all areas of the body. This is the kind of thing that drives conventional clinicians completely batty. It's impossible to really even figure out what's being said there. So when you look at the actual literature, as I've said, there was a review that was published in the American Heart Journal in 2000 that looked at 22 studies that had from 1 to 3,000 patients, no control groups, mostly reporting EDTA as effective. There was a systematic review published in 2005 that found 7 randomized trials. 2 studies with a total of 19 patients found some benefit. 3 studies with a total of 269 patients found no benefit. So they concluded that use of EDTA was not supported by highest quality evidence, but they were encouraged that the NIH is funding a large, simple trial. And given the widespread use of EDTA chelation therapy, assessment of safety is crucial. In the meantime, mainstream medicine was lining up against the use of chelation in ever more vociferous terms. So AMA, ACC, AHA, American Osteopathic Association, American Academy of Family Physicians all came out during the 1980s and early 1990s with strong anti-chelation statements and positions. One example of that is this AHA opposition paper, which basically cites as their motivation for taking their position that they were concerned that patients would be tricked, essentially, into displacing more effective but perhaps less glamorous therapies for this very hyped and seemingly ineffective chelation. And so they were concerned from that point of view that it would harm patient health if these assertions were left unchallenged. On the other hand, the chelation guys felt that they were being persecuted by conventional medical practitioners, that they'd been harassed, vilified, smeared, pushed out of practice by conventional medical societies because they have a different point of view. So, you know, a relativist perspective on truth and who owns it. So in that setting, we come up to the things that motivate the use of CAM, and you've seen some of these statistics perhaps before, but complementary and alternative medicine is used by at least 1/3 of the US population, if not more. 1/3 will admit to using it. Now, of course, it's a wide variety of things that are considered to be complementary and alternative medicine, including massage, deep breathing, meditation, yoga, as well as acupuncture, homeopathy, chelation, a variety of different things fall under that rubric. The National Center for Complementary and Alternative Medicine was established by the NIH in 1998 to perform rigorous scientific investigations of these therapies. It was considered to be in the public health interest to try to figure out whether some of this stuff actually worked or not, and they were willing to put some money into figuring that out. And the motivation for the Tac Trial then, factoring in those characteristics, is stated by Dr. Gary Gibbons, who's the current director of NHLBI, although he wasn't in his position when this study was started, was that the use of chelation was growing substantially. They estimated up over 100,000 patients apparently per year getting this therapy in the United States, and no evidence as to safety or efficacy. And then it was, so they felt that that was sufficient reason to go ahead and do a clinical trial, notwithstanding the fact that the understanding of how the thing might possibly work was certainly far from perfect. The Tact Timeline you can see in this slide. So there's an RFA request for applications released by NCAM and NHLBI in April of 2001. We put in our application, we got picked. TACT was funded as a cooperative agreement in August of 2002. There was an investigational new drug application obtained from the FDA in 2003 to use the chelation therapy because it hadn't been approved in this country for this purpose. And the first patient was randomized in September of 2003. The last patient was randomized in October of 2010. The results were presented initially at the AHA meeting last November. The second portion of the major results were presented this spring, last month actually, at the ACC, and the first major paper out of TACT came out last month. So, and this is a large, major NHLBI clinical trial with all the accoutrements and pieces that you would expect in such a trial. This is not a bunch of guys in a garage sort of trying to figure out how to do some stuff. You know, so we had the whole thing. But in 2008, a group of people who were self-appointed protectors of something decided to target the TACT study as being offensive to, I don't know, to them mostly. And they were assisted in this by Dr. George Lundberg, who was the ex-editor-in-chief of the JAMA, and who I guess was feeling a little bit neglected in his new position, wanted some attention. So they published an 80-some-odd page diatribe that was allegedly peer-reviewed that said that the TACT is unethical, dangerous, pointless, and wasteful, and it should be abandoned. And that was, you know, a little upsetting for those of us that were doing this study. But on top of that, they actually sent a letter to the OHRP alleging a whole bunch of improprieties, which the OHRP, then not knowing what to do, sent a letter to the trial leadership. The trial leadership, then not knowing exactly what to do, decided that we had to voluntarily suspend all of the trial activities until we could figure out what was going on. The trial was resumed study operations 1 week later. In terms of following and treating the patients already enrolled, new enrollments were put voluntarily on hold until we could finish the response to OHRP. That was sent by the TACT leadership to OHRP in December of 2008. New enrollments were restarted at that time. A minor edit to the consent form was improved by the IRBs that were involved in the study. October 2009, OHRP sends the final letter back to the TACT investigators. No need for further involvement by OHRP. In other words, much ado about nothing. But it certainly spent a lot of time doing nothing here. Dr. Lee is going to take over and get you through the trial results.
And so it's on. Well, thank you, Dan. That was really a terrific background sketch of some of the events that have transpired over the past decade. Are my slides here?
Yeah, you should be able to just go on.
This is really quite a remarkable saga. A decade, over a decade long effort. And of course, over that period of time, there have been really very important contributions from many different individuals. A number who are in this room today. There are a number who have since left the DCRI. In fact, I was thinking about generating a survival curve of everyone who's touched this trial at the DCRI, but chose not to do that. But I do want to mention, actually, and I've listed here, the numerous people at the DCRI who have been a part of the Tac Trial. Clinical operations, I want to particularly recognize the important work of Sandy Turt-Ulich, Lindsay Lamb, Wanda Parker, who I believe is in the audience, and several others. I see Brian here, a great team of people in ClinOps statistics. I particularly want to highlight Lauren Lindblad, who's been with the study from its inception and who was just a terrific support throughout the trial. We had our CEC group, our finance person, the safety group, Dan and his equal team, along with Nancy and others, and then all of these people associated with data management over the years is quite remarkable. And this is not all of the names, because I didn't list the names of people who are no longer at the DCRI. So you can see that this has been a huge effort involving a lot of people. And the one thing that I would say is that TACT certainly could not have been completed without the terrific and important efforts of all of these people. Every person's contribution has been very important to the trial, and so I want to offer my expression of thanks and appreciation to everyone who's been a part of this study over the past 10 plus years. So just to carry you a little bit further beyond the slide that Dan showed you with the flags highlighting. Oh, and I see Lauren Lytle back there. Lauren deserves to be mentioned. He was really one of the leaders of our data management team. So thanks to you, Lauren, and everyone else in data management. So after the final patient was enrolled in late 2010, and follow-up was then completed in late 2011. You might be asking the question, well, what did you do next? What happened? Because the results were not presented until the AHA in 2012. So what happened was that during the first 6 months or so, 6 or 7 months of 2012, there was extensive additional auditing that was performed at many of the TACT sites to make certain that all of the regulatory documents, particularly at the high enrolling centers, make certain all of the regulatory documents, everything was in order, that selected data checks were made in order to have the information in the study as pristine as possible. The database was finally locked then in August, the second week of August in 2012, and the final analysis was undertaken then, and the results presented at the AHA in November. The vitamin results presented at the ACC just a month ago, and then just within the past 10 days or so, the primary chelation results were published in JAMA. This is just a shot of the first page of that JAMA publication. I'm going to say a few things about the study design, description of the design and the various details that went into the study have been published, believe it or not, in the American Heart Journal. And so you can refer to this particular publication for greater detail than we have time to go in today. But TACT was designed as a factorial trial, what we call a 2x2 factorial trial, meaning there were 2 interventions that were being studied. And the objective, the intent, was that we wanted to study certainly the effects of chelation therapy, but chelation practitioners often use a high dose of vitamin and mineral therapy to accompany the chelation infusions. And so if the use of high-dose vitamins and mineral therapy was just haphazard, so to speak, in treating the patients, this could have been potentially a confounding factor that would have made the interpretation of the results more complex and more difficult. So in designing the trial, we decided to study both of those factors, namely the chelation factor as well as the use of high-dose vitamins, and control its use by randomization. So the table you see here is just a schematic of the 2x2 table. If you look actually at the if you look at the columns, the first two columns, you can see that those both received active chelation therapy. The two columns on the right received placebo. So the primary assessment of chelation therapy was to compare the columns in this 2x2 factorial design, whereas the primary comparison of the vitamins was to compare the two rows of this 2x2 factorial design. I just want to say also that the chelation infusions was not a trivial or an easy treatment necessarily. It involved 40 infusions, each lasting at least 3 hours, some of them longer than that. So there were 30 weekly infusions. Patients had to come back every week and spend 3 to 4 hours undergoing a chelation infusion. The last 10 infusions were more maintenance type infusions that were given 2 to 8 weeks apart. But all of the treatments were completely double-blind. There was an active chelation infusion, a placebo chelation infusion. There were the active high-dose vitamins and placebo high-dose vitamins, all of which was double-blind. And this is discussed at length in the design paper. The patient population in Tact, the primary parameters I think useful to remember are that these were patients that were 50 years of age or older. They had all experienced a myocardial infarction. The infarct had to be at least 6 months prior to their enrollment in the trial, and they had to have reasonable renal function with a creatinine less than or equal to 2 milligrams per deciliter. A few other enrollment criteria, but those are the main ones. The endpoints, the primary endpoint was a composite endpoint consisting of death, MI, stroke, coronary revascularization, or hospitalization for angina. So this is a composite of several different components. Obviously, some of those components are more important or perhaps more serious outcomes than others, but it's the primary endpoint was this composite. We had a key secondary endpoint of cardiovascular death, MI, and stroke, a set of what one might consider to be more hard or more objective endpoints. There were other secondary endpoints as well. The quality of life that was carried, the assessments that were carried out under the leadership of Dan and his team, and the cost and cost effectiveness, which has yet to be completed. The quality of life information was, for the chelation comparison, was presented at the AHA along with the primary clinical results. Just to tell you a little bit about the sample size and power calculations that went into the study design. The event rate that was assumed the time we designed the trial in the placebo patients was that patients would experience a 20% rate of one or more of these events after 2.5 years of follow-up. The 2.5 years is what we originally anticipated would be the average duration of follow-up of these patients. We wanted to have decent power for detecting a 25% reduction in the active arm. We planned for a 3-year enrollment period, a 1-year minimum follow-up, and patients were randomized in equal proportions to all 4 cells of this 2x2 factorial design. We were using an alpha of 0.5, and the goal for the power was to have at least 85% power. We also suitably accounted for some degree of non-compliance with the infusions and some loss to follow-up of the patients. The target sample size, as originally designed, was close to 2,400 patients. Now, there were some adjustments to the sample size that occurred over time. I want to emphasize that these adaptations or these adjustments were all made without knowledge of the accumulating data. So the accumulating treatment effect played no role in the adjustments to the sample size. The first adjustment occurred back in 2006 in a protocol amendment that was made at that time. The sample size was adjusted downward to 1,950 patients. The recruitment period was extended to 4.5 years. The minimum follow-up of 1 year was maintained. The other assumptions were unchanged, and these modifications would enable us to preserve the level of power that was desired. There was a second sample size adjustment in July of 2009, where the sample size was adjusted downward to 1,700 patients. The follow-up was again extended to preserve the desired level of power. And as a result of these different adjustments, the enrollment period then was extended over a period of 7 years, and the average follow-up of the patients would be approximately 5 years rather than the 2.5 years as the study was originally conceived. The analysis, some of the key ingredients to the statistical analysis plan, which was all pre-specified well in advance, was that the treatment comparisons would be performed using an intent to treat approach, two-sided testing with extensive use of confidence intervals. The log-rank test was to be used for the statistical assessments of the treatment effects. Interim monitoring was accomplished using the flexible alpha spending function approach that was developed by our friend Tom Fleming and Peter O'Brien with Dave Demetz and Gordon Land, the alpha spending function approach using O'Brien-Fleming type monitoring boundaries. Because of the multiple reviews of the data by the DSMB due to the protracted length of this study, the final level of significance that was required for the primary endpoint was 0.036. And we also performed a number of sensitivity analyses, which I'll describe later. So here are the primary results. The enrollment, the final enrollment was 1,708 patients. These patients were all enrolled in the U.S. and Canada. We had 119 sites in the U.S., 15 in Canada, 134 sites, and close to 90%, 89% of the patients were enrolled in the U.S. and 11% in Canada. The median duration of follow-up overall was 55 months. This amounted to over 6,000 patient years of follow-up in the study. The baseline characteristics, just briefly, the median age of the patients was about 65 years. Body mass index, the median value was 30. We had 18% females. There were a little over 30% of the patients with diabetes. This percent, 83% of these patients had undergone a prior coronary revascularization. So a very high percentage of the patients had been revascularized previously. Patients were generally on reasonably good background evidence-based therapy, over 70% on statins, over 70% on beta blockers, and over 80% on aspirin. In terms of the infusions themselves, there were a total of 55,222 infusions administered in this trial, which I think is really quite a remarkable number. 65% of the patients completed all 40 of the infusions, and just over 3/4 of the patients completed at least this intensive phase of 30 infusions. You can imagine that once patients got started with this infusion regimen, having to go in every week, spend 3 or 4 hours, some of these patients got tired and did not, they just didn't feel like they had the time to invest or to devote to this. There are a variety of reasons why some patients did not complete the entire regimen of 40 infusions. But I think that it was admirable that we had as many of these patients as we did that completed the number of infusions that were completed. Now, there were also challenges in the trial that I want to mention with withdrawal of consent. Many of these patients who opted to withdraw from completing the infusions also opted to withdraw completely from the study. So this is a much higher percent, 17%, than we typically see in these clinical trials. And this was very alarming to me and others. But 17% of the patients withdrew consent at some point during the course of their follow-up. And there was a differential withdrawal of follow-up, 14% in the chelation arm and 20%, a higher percent, in the placebo arm, a significantly higher percentage. The median duration of follow-up of these consent withdrawal patients was 10 months. 18% of those patients, however, had an endpoint prior to their withdrawal from the study, so it's not as if we didn't get any information, any important information from those patients. And at the end of the study, we did a careful search of registries, the National Death Index, etc., to assess the vital status of all of the patients, including these patients who had withdrawn consent. There were 22 patients who, for various reasons, were lost to follow-up during the course of the trial. So here's the primary results. This is the Kaplan-Meier curve for the placebo arm in the trial. This is based on Kaplan-Meier calculation using time until the occurrence of the first event in this composite consisting of several different possible outcome events. 38% event rate at 5 years in the placebo arm. And this is in contrast to the patients who underwent chelation therapy. 33% event rate, so there was an absolute difference of 5% at 5 years in the event rates, and the relative reduction, the hazard ratio was 0.82, so about an 18% relative reduction with a p-value of 0.035. I want to just show you some of the results of the components of the primary endpoint as well. The first row here is just what I've shown you. The second row is mortality by itself. So there was not much, there was not any real evidence that chelation therapy was reducing mortality, hazard ratio of 0.93. But in terms of some of the other components, it's interesting that the hazard ratio is in the 0.7, 0.8 range for MI, for stroke, for coronary revascularization, hospitalization for angina. Now, the number of events for these components is smaller, and certainly the study was not powered to detect statistical differences in those individual components. It's interesting that one of the most frequently occurring events was coronary revascularization, and there was quite a substantial reduction in the amount of revascularization in the patients who underwent chelation therapy. So just a word or two about the withdrawn or lost patients. As I said, there were significantly more patients in the placebo arm who withdrew consent compared to the active arm. We may therefore be missing outcome events in this trial because of these withdrawals from the study. But when we look at the difference, the fact that there were more withdrawals in the placebo arm compared to the active arm, if we're missing data, it's likely that we're missing more events in the placebo arm. And if we had all of the data, it would likely make the treatment effect even more significant. But we performed some sensitivity analyses to examine the robustness of the results, how these withdrawals might have affected the results. Just briefly, we did a number of different analyses where we imputed event rates under multiple different scenarios. And this is really well described in the online supplement to the JAMA paper for those of you who are interested in more detail about this particular aspect of the analysis. But the result of the analysis was that the significance of the trial was preserved for plausible scenarios as well as many implausible scenarios where the event rate among the withdrawn patients in the active arm was somewhat higher, as much as 25% higher than the event rate among the withdrawn patients in the placebo arm. So as a result of these extensive sensitivity analyses, we concluded that the results appeared to be robust in terms of the statistical significance. I won't go into detail on subgroup results. There were some interesting results here in that patients with an anterior myocardial infarction had a stronger result. There was a significant interaction p-value, as well as patients who had diabetes. you can see that in diabetics, actually the hazard ratio was 0.61, an impressive nearly 40% reduction in patients with diabetes. We have to be cautious about the interpretation of these subgroups, but you'll note that the hazard ratios are mainly to the left of unity in the direction of a benefit for chelation therapy. This I want to just mention because looking at the complementary and alternative medicine sites, although the interaction is not significant, you can see that the non-CAM sites, which were largely cardiology practices, had an even better outcome than did the CAM sites, where you would expect potentially there to be more of a bias. So I'm going to skip over this and just point to the bottom line that safety was not an issue in this particular trial. If anything, there were more adverse events in the placebo arm than in the active arm. And let me come to these conclusions later. And show you just very briefly, I wish we had another hour here because the data are really so fascinating. But I want to give you a flavor of the vitamin comparisons now, if we could. You know, the comparison of the two rows of this factorial design. These vitamins were not just small pills. They were horse pills. And so patients were to take 6 of these a day, three tablets twice a day for the duration of the study. And again, they were double-blind, and just taking those vitamins and sticking with that over the course of the trial was a challenge. But 3/4 the patients took those 6 large vitamins for at least a year. A number of them refused to continue. The median duration of vitamin treatment was nearly 3 years. And there was no difference in the discontinuation rate in active versus placebo vitamins. This is again, the placebo curve, the vitamin placebo curve. And here's the active high-dose vitamin curve. So here you see that there was only a 3% absolute reduction at 60 months, and the relative reduction was less. It was not statistically significant, an 11% relative reduction. Interestingly, the subgroup that was interesting here with the vitamin comparisons, where there was a significant interaction, was in patients with statin therapy. Patients who were not on statins seemed to do better with this vitamin therapy, nearly a 40% reduction in primary events. So just to augment this by showing you results in all 4 of the cells of this factorial design, this is the group that got placebo infusions and placebo vitamins. This is the group that got placebo infusions and high-dose vitamins. This is the group that got active chelation but placebo vitamins. And this is the group that got active, both the active chelation therapy and the active vitamins. And I'm highlighting those particular curves, the placebo cell versus the cell in which the patients received active therapies, both from chelation as well as vitamins. And you can see that the hazard ratio comparing these curves is 0.74. The p-value was 0.016. So in summary, the chelation therapy reduced the composite outcome by 18%, which was statistically significant. The high-dose oral vitamins reduced the composite outcome by 11%, which was not statistically significant. And there's certainly a caveat here. Compliance with the high-dose vitamin regimen was challenging, as I've already said. If all of the patients had taken all of the vitamins over the nearly 5 years of follow-up, it's interesting to conjecture what the results of the vitamin comparisons might have been. There was no interaction between chelation therapy and high-dose vitamins. The effect of the chelation therapy was consistent whether the patients received the vitamins or not. When combined with the chelation therapy, the small vitamin benefit was additive, and the combined effect when we compare active-active versus placebo-placebo in this factorial design reflected a significant 26% reduction. We need more research to understand the therapeutic mechanisms of this interesting intervention, and hopefully TACT will stimulate that further needed research. So those are highlights of the results. I know we're running out of time, but Dan is going to share with you some interesting things that have happened since these results were reported.
Thanks, Kerry. So mindful of the hour, I will try to go through a few things quickly. Basically, so this is the reaction. What was the reaction of our colleagues when the TACT results were first presented at the AHA? It was a big headache, as Dr. Fuster shows here. He has this thing on the heart.org, which is the cardiology show, where he gets a bunch of experts around to talk about the trials that were presented, and they were all kind of speechless, basically. Valentine's comment was the whole list of statistics was really very borderline, which I have no idea what he means by that. But if you imagine him delivering it in his Catalan accent, it sounds very good. Lars Valentine said, "I think the results are provocative, and of course we like to dismiss them because it's so surprising," which is a very insightful comment. Rita Redberg, "I don't get the whole thing. There should be a credible scientific basis for your hypothesis." Clyde Yancey, "We need to pause, really, to put this into context. My own feeling is we should pause indefinitely." Bob Harrington, "If this is real, it might be worth studying." And Noel Barry Mertz says, "As a scientist, I don't believe it. We need to see 3 trials that are consistent." 3 trials, wow. Anyhow, this news story pretty much summarized the feeling of many at the AHA, which is dismay. And the New York Times, quoting Dr. Nissen at the Cleveland Clinic, said it is a potential public health catastrophe. Nissen said the study was fatally flawed. If people got the mistaken idea from the study that chelation was beneficial, it would be a disaster. Echoing back to the earlier sense that I think he's trying to say is that it would displace all the good stuff that we've discovered in the meantime, and that would be a bad thing for patients. Nonetheless, and despite the efforts of some to prevent it, the TACT was published by JAMA, as you've heard, last month. And the editors went to the unusual length of explaining themselves in print as to why they did that, anticipating that they were going to get a little bit of blowback. So they said the decision to publish was related to the investigators, who include one of the most preeminent cardiovascular researchers, and I think we need to acknowledge Tony Lamas, who was truly a visionary and statesman-like leader in one of the most difficult clinical trials I've ever seen. and one of the most respected statisticians, Dr. Kerry Lee. So Dr. Lee was in part responsible for JAMA's deciding that this was not just a bunch of BS, because it came from Dr. Lee. Although many physicians have biases about chelation therapy and other CAM therapies, the scientific process should prevail in providing evidence about these therapies. Reports of rigorous investigation should not be censored because of pre-existing ideological positions. Thank you. However, Dr. Nissen was not deterred. Given the numerous concerns with this trial, including missing data, potential investigator or patient unmasking, use of subjective endpoints, and intentional unblinding of the sponsor, NHLBI he's talking about, the results cannot be accepted and do not demonstrate a benefit for chelation therapy. So Dr. Nissen is going to go up against Dr. Lee, basically. You know, it's like you're going to play one-on-one basketball with Michael Jordan, and you don't have any shots. All you got is trash talking, basically. You know, you can't take a jump shot. You don't have the three-pointer. The sky hook doesn't exist. It's a silly thing to do. So for all those TAC naysayers, I think there's some ancient but relevant advice that can be obtained from Matthew, chapter 7, verse 1 through 5, where it says, "First cast out the beam out of thy own eye, and then thou shalt see clearly to cast out the mote of thy brother's eye." So while they're busy trying to help us with our motes, we're thinking about those beams. So fortunately, it was very enlightening, I think, to see the response of our colleagues in the cardiovascular community to this very challenging clinical trial. I think several of our colleagues really deserve a lot of credit for stepping up. Carlin Krumholtz was blogging about the Tac trial and has taken, I think, a very open-minded and accepting perspective. He says, "I'm glad that we're subjecting popular but out of the mainstream practices to rigorous study. If I endorse that course, I cannot ignore the evidence because it goes against what I expected. The irony is if a drug manufacturer had gotten this result, they would have celebrated." And that's true. Dr. Topol, daring to challenge dogma. He says the trial was imperfect, but it was the trial was imperfect, but it was a daring step toward a better understanding. Seemed like a crazy notion in 2003 when the NIH invested the money in it. But the consistent and notable reduction of a spectrum of clinical endpoints indicates that maybe something important and controversial is happening. So, but there is a lot of recurring criticism that have been raised, and I just want to mention just a couple of things here at the end. The results are highly unexpected. Yes, of course, but what's that mean, really? Sanjay Kaul at UCLA, or Cedars-Sinai, did a sort of mini Bayesian analysis on the hazard ratio and calculated that the hazard ratio with a skeptical prior would go from 0.82 to 0.87. It still makes the results of chelation to be more likely than not positive, if you want to put it in that perspective. So even with a very skeptical prior, you still have to give some credit to the hypothesis after you're finished. The high rate of dropouts that you've heard about from Kerry already, it actually creates a conservative bias against chelation. Alleged misbehavior of the CAM sites, it actually, you saw that the CAM sites had less of a treatment effect than the non-CAM sites. So why don't you actually read the paper before you put those criticisms out there? The results are marginally significant. The p-value reflects an element of precision, not the effect size. The composite endpoint dominated by revascularization events. Again, the effect size is comparable for all the events. Let's read the paper before we make these comments. So, Dr. Tukey, who's one of the famous and preeminent statisticians of the last century, says it's not always easy to recognize what the data are saying to us. The understanding process is clearly a journey, not a destination. Thanks for your attention.
So Dan and Kerry, thank you for that, getting us a little closer to the truth, whatever that is. The hour's late. I have one question, and then maybe we'll take one or two from the audience. So, what are you recommending to your patients with regard to chelation therapy?
I think this has come up a lot, and the question is, so you've seen the literature. So basically, there's a couple of studies from the 50s and the early 60s, and then there's basically nothing. There's a few very small trials that don't really tell us anything, and then we've got a large trial here. So we really don't have, a great deal of understanding. We used a very complex intervention, this combination of all these vitamins and this chelation thing with 40 infusions. I don't think we really understand it all yet. It's a very difficult treatment. This is not something you'd undertake lightly to do. 40 infusions is essentially like going for chemotherapy in a way. You're coming back repeatedly, or dialysis or something. It's very challenging. We don't know about whether the dose is right. We don't know how, the vitamins have to be continued forever. I think it's not an emergency that we have to use this right away. And it certainly makes sense to get more information before we decide how to apply this clinically. If this is the right thing, maybe this is just telling us some interesting mechanistic information about atherosclerosis. There's some element of the disease that we have heretofore been ignoring or been unaware of that is amenable to some sort of therapeutic intervention, and maybe we should figure out what that is. And that may be the main message of TACT.
I wish we did have another hour because there's so much that we could talk about. But I want to follow up on John's question that rather than for the individual, I think the question of what should be done in the way of research next. And it seems like there are three options. Most of the pundits have said, forget about it and let this die a slow death and don't do any more research. You might argue you should do a very large, very simple trial without worrying about all those details. I still remember, the seminal moment for me was when we were at the FDA with the EPIC trial way back with abciximab. Clinical pharmacologists were going to vote it down, and Bob Temple asked the question, can you name the correct dose of any drug for cardiovascular disease? There were 3 minutes of absolute silence. And when no one could name the right dose of any drug, they went ahead and voted for approving this one because of the empirical evidence. Now, others would argue that you need to go completely the other direction and do basic science and explain why this may or may not work as a fundamental line of evidence. Which way should we go?
Well, of course, the practical problem is that you're finding the money to do a large clinical trial where there's no sponsor that's going to have a commercial interest in the outcome. And despite all the nice words that have come out of NHLBI lately, I'm having trouble seeing them in the current environment shelling out another 40 or 50 million to replicate this. So, in the ideal world, I think we would clearly want to do another large-scale clinical trial.
Why not just randomize to, forget the factorial, randomize to the treatment or not, and measure.
The new hypothesis is that vitamins and the high-dose vitamins and the chelation together versus placebo would be the thing you would try. I don't think you would need to do the factorial design anymore. There may be other elements that we would look at. Some of these subgroup issues may need to be drilled into further, but I think that you've got to, how do you figure anything out when you've got something unexpected, that you've discovered a therapeutic principle of something? I mean, look at all the drugs that we used that started off one place and ended up in some place completely different when we finally figured out what they were more useful for. There's a lot of drugs that we use didn't start where we now use them.
I can't resist asking, as a noted clinical investigator, how would you rate the use of percutaneous intervention for chronic coronary disease relative to chelation therapy?
Yeah, you've
the evidence is clear,
read my
isn't
new
it?
book about. Well, clearly, there's a lot of interesting. Those of us in conventional medicine also are subject to a lot of beliefs that drive our behavior, and conventional doctors also, even though think that they understand stuff, and that drives what they do, even though the evidence is often lacking. I mean, we've had a huge problem in coronary disease dissociating ourselves from the simple pathophysiologic model that if you open a coronary, somehow that's got to be beneficial when you look at the thing and it looks so stenosed, but yet there's huge amounts of empirical data to show that in many of those cases we're doing nothing for the patient except making them a better patient because they're going to come back and have a bunch more procedures to take care of the procedure that we did initially. And that some simple medical therapy is actually a better strategy for many of these folks than mechanical strategy. But I don't know if that was what you were getting at.
So just quickly, I'd just like to draw one analogy, not to percutaneous intervention this time, but to another study that came out nearly about the same time as yours, just a little after it in that other regional journal up north. And it was called the Mediterranean Diet. And I don't know how many people saw this study, but in fact, the treatment effect size is similar. It's a slightly larger study. They had big-time issues of crossover and dropout and other things that existed within that study. The study has been in every newspaper hailing the fact that we've now proven for sure that the Mediterranean diet saves lives and prevents events, and everybody should be adopting it. The editorial attached to that article said, we probably really never even needed to do this trial because we had all this observational data ahead of time that showed us that the Mediterranean diet worked. But we don't know exact amounts about dosages and how it might work. But we still, no matter what, we should absolutely adopt it, and this should be taken in national policies that we should be providing free olive oil and nuts to our populations. Interesting, distinction between two studies with almost the exact same results, based on just the degree to which we have presupposed ideas about which one works and which one doesn't.
Could I ask just a practical question? John asked the question what you're doing with your patients, and you said in the presentation that you needed an investigational new drug application because this drug is not approved. So what would be the opportunity to actually use this therapy in practice? What's the FDA's view as to whether consistent, systematic use would really be a violation of off-label use parameters?
The FDA asked all the manufacturers that were currently making disodium EDTA to stop making it. There's a calcium EDTA which is used for lead toxicity, which is still available, but the disodium version has been taken off the market. This happened during the trial. And so you can still get it in compounding pharmacies, but you can't get it from a pharmaceutical manufacturer right now. And again, the problem is who's going to go to the trouble of doing an NDA for chelation therapy. So it's an interesting problem right now.
Dr. Mark, would you expect the placebo effect of this therapy to be greater than conventional therapy? And if so, would you take that into consideration when interpreting these findings?
Well, the placebo effect is I guess that's an interesting question. The original data that I showed at the beginning of the talk was that suggested there was no treatment effect. They were looking at symptoms and there was no treatment benefit for 20 weeks. And then eventually something started to happen. So it's a little hard to see how that could fit if if it happens after you finish administering the therapy. It's hard to see that that's a placebo effect. Now that's for angina. Now we're not looking at angina, we're looking at stuff that happens to patients. It's harder to see how those kind of events, strokes, MIs, repeat revascularization could be, hospitalization for angina could be driven by somebody's belief. And we have some interesting data about what they thought they were getting, but we're not yet completed analyzing it. But I don't think at the moment that we see a placebo effect. A lot of patients in both groups thought they were getting the good stuff, basically.
So Dan and Kerry, the hour's late. Thank you very much. We'll schedule a follow-up. Have you for another hour to talk about the